Alright, in this unit we're going to be focusing on one of the six kingdoms that we looked at a couple weeks ago. We're going to be looking at the bacterial kingdom. Alright, if you remember in our activity with the six kingdoms, we know that there are two bacterial kingdoms, okay, the eubacteria and the archaebacteria. Then you have Protista, the fungi, Plantae, and Animalia. And we're going to be looking at the bacterial kingdoms today. So let's first get a glance into archaebacteria. We're not going to spend just a lot of time with them, but I do want to kind of go over the structure a little bit, let us know some of the components that make them up. If you remember from your research you did, your archaebacteria is also known as your extreme living bacteria. They're going to be found in places around the planet where there are not normal conditions as far as environmental conditions or some type of extreme or harsh condition, whether it be temperature, whether it be chemical composition, whatever it may be, that is causing that particular habitat to be uninhabitable except by certain types of bacteria, which are these archaebacteria, or they're also known as extremophiles. All right, so for this picture here, this is a picture um, of a hydrothermal vent down underneath the ocean, pretty deep underneath the ocean, but located near volcanic activity. And so what you have occurring at these hydrothermal vents is that there are gases highly toxic or highly high in temperature. These gases, a lot of sulfur oxides and nitric oxides, carbon oxides are being pumped from from these volcanic tubes or that branch off from the volcanic activity close to the ocean floor. They're being pumped up into the very ice cold ocean water. So when these gases hit the water, they form very toxic chemicals as a result. Very high superheated waters, and you get rare types of organisms, especially these extremophiles that can grow and live in these areas. You have areas that form acid pools, and you can get archaebacteria growing in those. You have certain lakes or bodies of water that are really highly concentrated in salts, to the point to where there's no life living in them at all except certain types of archaebacteria. Other places like icebergs and glaciers, you'll find certain types of archaebacteria that can live just in those extreme conditions. Now, as far as some characteristics of these archaebacteria, they are prokaryotes, so that means they are single-celled. They have a cell membrane. They have a cell membrane. They've got cytoplasm. They are going to have ribosomes that can make proteins. They're thought to be related or ancestors of mitochondria. All right, one of those organelles within our cell. Uh, they do have similarities close to mitochondria and chloroplast. They're known as the ancient bacteria, some of the first ones that propagated on the planet. Archaebacteria do not have a cell wall, or excuse me, they do not have peptidoglycan. Peptidoglycan is a polymer carbohydrate that kind of forms a mesh-like cell wall around normal bacteria. Well, archaebacteria don't have that particular carbohydrate that forms their cell walls. They do have cell walls. It's usually made of cellulose or a cellulose type substance. Um, they are heterotrophic, all right, so they are not autotrophic, and their main form of reproduction is going to be through binary fission or budding. And that's, remember, we talked about binary fission before. That's where they copy all of the materials within inside their body, and then they split in half. So they just make a copy of themselves and split in half. That's binary fission. That's their main form of reproduction. All right, now the other type of bacteria, and this is the one we're going to kind of focus on today because this type of bacteria is going to be known as our true bacteria. This is the common bacteria that actually is used to help life cycles. It's used to help those biological cycles for the natural functioning of life and the environment. The true bacteria is what's going to, or the eubacteria is what's going to be a part of that. Okay, so looking at some characteristics of our eubacteria, they're also prokaryotes, just like our ancient bacteria. Um, but the eubacteria, we think, might have been the start of organisms that gave rise to eukaryotic life. But they're single-celled. They also have cell membrane, cytoplasm, ribosomes for protein synthesis. Their cell walls are made of that peptidoglycan, okay, which is that mesh-like carbohydrate polymer that forms their particular cell wall. Where the archaebacteria doesn't use that polymer, it uses a cellulose type polymer. Our eubacteria, or our true bacteria, they can be photosynthetic, they can be chemosynthetic, or they can be heterotrophs. All right, so they can make their own food and energy through photosynthesis or chemosynthesis, or they could be heterotrophic. Whereas our archaebacteria is strictly heterotrophic; they're not autotrophs. The habitat for eubacteria is all the common types of environments, so no extreme environments. You're not going to find common bacteria where there's extreme temperatures, whether it be extreme cold or extreme hot temperatures, strong chemical concentrations. You're not going to find eubacteria there. Your eubacteria is going to be found in common places, like in a forest, in the desert, outside your door, on your door, in your house, on your phone. So all the common types of environments where there's normal temperatures, normal, steady for the most part, climates, that is where you're going to find their habitats. They're very diverse. Okay, there's over 5,000 species of different types of eubacteria, or the common type of bacteria. They can reproduce through binary fission, like archaebacteria can, but they can also reproduce through sexual reproduction, like using through conjugation or spore formation. All right, so looking at our true bacteria, our common bacteria, there are some basic shapes that they can form, or that their body shapes or body structures will take on. They can be spherical in shape, they can be rod shaped, or they can be curved in shape. So here are some pictures that go along with that. Your cocci bacteria are going to be spherical in shape. Your bacilli bacteria are going to be rod shaped, and out of all of these, most of your bacteria would probably, or the ones that we look at that are more common to what we'll use, would be bacilli. But there are just as many that could be cocci or spherical, or spirilla, which would be curved shaped. So make sure you're good with the scientific terms related to the shape of these bacteria. Here's the basic structure. All right, this is a bacillus, a rod-shaped structure of a bacteria. It does have genetic material, so it's DNA located in its nucleoid region. It doesn't have a nucleus. Remember, your bacteria, your prokaryotes, don't have membrane-bound organelles, so they don't have mitochondria, they don't have a nucleus, they don't have smooth ER, rough ER. It does have DNA, though, in its nucleoid central region. It does have ribosomes to help make protein. It has plasma, cytoplasm in it. It has a plasma membrane. It has that cell wall made of peptidoglycan, that carbohydrate polymer. This is the basic structure of all true bacteria. They're going to have all of these characteristics. Now, the reproduction methods, let's look at those briefly. They can go through binary fission, and we've talked about this, where they just copy or replicate what's inside their organism, their cell. So they'll copy their DNA, they'll make sure they have extra ribosomes and cytoplasm, and then their cell membrane and cell wall will pinch in half and actually cause the grown or enlarged bacteria to split in half and form two. That's binary fission. It's asexual. But they can also go through reproduction, sexual reproduction. One way would be through conjugation. These are going to be your example of conjugation for your cocci. Okay, they're going to connect using bridges, protein bridges. They'll use proteins to form bridges between two different bacteria. What they'll do when that happens is they will transfer some of their DNA to that other bacterial cell. Once that happens, the bridge is broken. The conjugation process is over, but then those new separate pieces of DNA will be incorporated into that other bacterial cell. So it's increasing diversification with the bacterial DNA through conjugation. And then there's spore formation. A lot of bacteria reproduce through spore formation. And what spore formation is really it's almost like making a seed, if you will. What's going to happen is the bacteria is going to form a really tough protein structure with inside the bacteria. It's going to put some of its cytoplasm, some of its DNA into this protein structure that they formed. Okay, it's going to form something called an endospore, almost like a seed that's going to carry, or it's going to hold some of its DNA and cytoplasm. This endospore is very tough, very resilient. It can withstand harsh conditions, chemicals, um, radiation, loss of water. It can last for millions of years like this, but eventually this endospore, once it finds the right growing conditions, it'll activate and begin to grow and begin to reproduce new bacteria. So that's what spore formation is. It's just kind of when it forms this endospore, it's almost giving it a chance to survive when conditions aren't favorable.